These are genetically modified mosquitoes, but they are not like any genetically engineered organism ever created before. They are designed to spread their genetic mutation in the wild and hopefully someday eradicate the mosquitoes that transmit malaria, one of the deadliest diseases on the planet. Scientists are experimenting with these mosquitoes in a lab that is designed to make sure none of the insects escape. The lab is in Italy because even if they do escape, these mosquitoes couldn't survive in this climate.
Okay, so, one moment.
Ruth Mueller runs the lab. She agreed to give me exclusive access to the lab on the very day she launched this controversial experiment. She showed me the mosquitoes in various stages of their development.
And all these microscopic structures, you can find this beauty in every of the organisms on Earth. So you see the eyes, right and left. This,
under the microscope, the modified insect's eyes glow yellow when they are lit up with a laser. The mosquitoes were created using the powerful new gene editing technique known as CRISPR, to carry a sequence of DNA called a gene drive. The gene drive empowers the mosquitoes to spread their mutation incredibly fast through their entire species. The mutation turns all the female mosquitoes into mutants, half female, half male. Their mouths are male so they can't bite and spread the malaria parasite. Their reproductive organs are deformed so they can't lay eggs.
Everything is bad, so it's not that we are against mosquitoes and we don't want to eradicate them, we want to control them, and this is quite a big difference.
She takes me into the most secure part of the lab. This special chamber is where scientists are, for the first time, releasing large numbers of these CRISPR gene-drive mosquitoes into cages filled with hundreds of natural mosquitoes of the same species.
Yes, it's a big moment. So it's the first gene drive release into a, let's say, near natural population. Two things will happen. So first of all, we make the gene drive release. So we put a specific number of gene drive mosquitoes in 2 of the 6 cages.
Everything in the chamber of large cages is carefully controlled to mimic the mosquito's habitat in sub-Saharan Africa. It's hot and humid.
So Tanya, our chief technician, she is now putting a vessel with all this gene drive pupa into the large cage.
The hope is that if these mosquitoes are someday released in Africa, they would mate with the main species of mosquitoes that spreads malaria, quickly transmit their modification through local populations, and help wipe out malaria. But these mosquitoes are really controversial. Critics fear they could wreak havoc with the environment if they were ever released into the wild. That's why Mueller and her colleagues are being so careful and only releasing them inside this highly secure lab.
Now put the small devices with this heated blood into the cage.
Heated cow blood to feed the mosquitoes. This is the second step after dozens of immature CRISPR gene-drive mosquitoes have been placed into the cages.
Yes, we want to give them space to see the natural behavior, so we mirror as best as we can the African environmental conditions.
To see their natural behavior, a computer controls the intensity and color of the light to mimic sunrise and sunset every day.
Yes, this is the beginning of the sunset, so they are going to sleep very soon.
Mimicking sunset is crucial because this is when the male mosquitoes start their mating dance and attract females to spread their sterilizing mutation.
I really hope that we can contribute to the eradication of malaria in the long term, so this is the final goal we have.
But Mueller knows how worried people are about these mosquitoes, so scientists are planning years of experiments to prove the mosquitoes work and would be safe to set free into the world.